Hello friends, today we are going to talk about hemostasis, hemorrhagic disorder and thrombosis. So, let's discuss introduction first. Hemostasis can be defined simply as the process by which blood clots form at the site of vascular injury. Hemostasis is essential for life and is deranged to varying degree in a broad range of disorders, which can be divided into two groups. In hemorrhagic disorders characterized by excessive bleeding, hemostatic mechanisms are either blunted or insufficient to prevent blood loss. By contrast, in thrombotic disorder, blood clots, often referred to as thrombi, form within intact blood vessel or within the chamber of heart. As in discussed in chapter 11 and 12, thrombosis has a central role in the most common and clinically important form of cardiovascular disease. So, what is hemorrhagic disorder? It is characterized by excessive bleeding, in which hemostatic mechanism is insufficient to prevent blood loss. Whereas in thrombotic disorder, their blood clot is formed within the intact blood vessel. Although useful, it must be recognized that this division between bleeding and thrombotic disorder sometimes break down, in that generalized activation of clotting sometimes paradoxically produce bleeding due to the consumption of coagulation factor as in disseminated intravascular coagulation (DIC). To provide context for understanding disorders of bleeding and clotting, this discussion begins with normal hemostasis, focusing on the contribution of platelets, coagulation factors, and endothelium. So let's discuss about normal hemostasis. We will discuss about this in today's video. Hemostasis is a precisely orchestrated process involving platelet, clotting factor, and endothelium. These three components are required, and we will discuss about these three. That occurs at the site of vascular injury and culminates in the formation of blood clot, which serves to prevent or limit the extent of bleeding. Let's see it again. Hemostasis is a precisely orchestrated process involving platelet, clotting factor, and endothelium that occurs at the site of vascular injury and culminates in the formation of a blood clot, which serves to prevent or limit the extent of bleeding. The general sequence of events leading to hemostasis at the site of vascular injury is shown in figure. The first of all, arteriolar vasoconstriction. Arteriolar vasoconstriction occurs immediately and markedly reduces blood flow to the injured area. It is mediated by a reflex neurogenic mechanism and may be augmented by the local secretion factors such as endothelin or potent endothelium-derived vasoconstrictor. So it will be augmented by endothelin. And it is mediated by reflex neurogenic mechanism. This effect is transient, however, and bleeding will resume if not for activation of platelet and coagulation factors. You can see here figure. This is transient vasoconstriction. Due to reflex with the reflex vasoconstriction, endothelin release augment vasoconstriction. This is site of injury. After this is normal. After vascular injury, neurohumoral factors induce transient vasoconstriction. Here, primary hemostasis, the formation of platelet plaque. This is second. After arterial transient vasoconstriction, now there will be primary hemostasis, the formation of platelet plaque. So, what is primary hemostasis? It is the process in which there will be formation of platelet plaque. Disruption of the endothelium exposes the subendothelial bone Willebrand factors BWF and collagen, which promote platelet adherence and activation. Activation of platelet results in dramatic shape change from small rounded disc to flat plate with spiky protrusion that markedly increased surface area, as well as the release of secretory granule. So in activation of platelet, two things will be occurring. First one is there will be dramatic change in shape, and and there will be release of secretory granule from platelet. This platelet adherence and activation is promoted by bone element factor and collagen. Within few minutes, the secreted products require additional platelet that undergo aggregation to form a primary hemostatic plug. So you can see here. We will discuss a detailed mechanism. We will discuss later. Platelet bind via glycoprotein 1b, GP1b to bone Willebrand factor on exposed extracellular matrix ECM and are activated undergoing a shape change and granule release. Released adenosine diphosphate ADP and thromboxane A2 induce additional platelet aggregation through platelet GP1B to 3A receptor binding to fibrinogen and form the primary hemostatic plug. So, what is the process? First of all, platelet bind via platelet glycoprotein 1B receptor bind to bone Willebrand factor, subendothelial bone Willebrand factor, and th- glycoprotein 2B3A bind to fibrinogen. You can see a detailed process. This is endothelium basement membrane. This is subendothelial bone Willebrand factor. There will be platelet addition to subendothelial factor. There will be shape change. Then thirdly, there will be granule release. Then there will be more additional recruitment and aggregation of hemo. More platelet will be aggregated to form primary hemostatic plug. Third, secondary hemostasis, deposition of fibrin. Vascular injury exposes tissue factor at the site of injury. Tissue factor is a membrane-bound procoagulant glycoprotein that is normally expressed by subendothelial cell in the vessel wall, such as smooth muscle cell and fibroblast. Tissue factor binds and activates factor 7, setting in motion a cascade of reaction that culminates in thrombin generation. Thrombin cleaves circulating fibrinogen into insoluble fibrin, creating a fibrin meshwork, and also is a potent activator of platelets, leading to additional platelet aggregation at the site of injury. This sequence, referred to as secondary hemostasis, consolidates the initial platelet plug. See here. Local activation of coagulation cascade involving tissue factor, and platelet phospholipid result in fibrin polymerization, cementing the platelets into a definitive secondary hemostatic plug. You can see here tissue factor will be released. It will be phospholipid coagulation factor complex. There will be thrombin activation, fibrin polymerization, and there will be secondary hemostasis. And fourth and last is clot stabilization and resorption. Polymerized fibrin and platelet aggregates undergo contraction to form a solid permanent plug that prevents further hemorrhage. At this stage, counter-regulatory mechanism tissue plasminogen activator (TPA) made by endothelial cell are set into motion that limit clotting to the site of injury and eventually lead to clot resorption and tissue repair. Counter-regulatory mechanism mediated by tissue plasminogen activator (TPA) of fibrinolytic product and thrombomodulin confine the hemostatic process to site of injury. Release of TPA fibrinolysis thrombomodulin blocks coagulation cascade trapped near trap cell fibrin polymerization. And there is new addition of 51 paragraph in latest 10th edition. It should be emphasized that its endothelial cells are the central regulator of hemostasis. The balance between the antithrombic and prothrombic activities of endothelium determines whether thrombus formation, propagation, or dissolution occur. Normal endothelial cells express a variety of anticoagulant factors that inhibits platelet aggregation and coagulation and promote fibrinolysis. After injury or activation, however, this balance shifts and endothelial cells acquire numerous procoagulant activities activation of platelets and clotting factors. Beside trauma, endothelium can be activated by microbial pathogen, hemodynamic forces, and pro-inflammatory mediators. Now we will describe roles of platelet coagulation factor and endothelium in hemostasis in greater detail following the scheme illustrated in figure 4.4. We have already seen figure 4.4. Already we have seen these are this is figure 4.4. We will see the detailed mechanism now. So we will talk about platelet in this video and about other coagulation factor and endothelium in next video. Platelets play a critical role in hemostasis by forming the primary plug that initially seals vascular defect and by providing a surface that binds and concentrates activated coagulation factor. Platelets are disc-shaped or nucleated cell fragments that are shed from megakaryocytes in a bone marrow into the bloodstream. Their function depends on several glycoprotein receptors, a contractile cytoskeleton, and two types of cytoplasmic granules. Alpha granules have the addition from a molecule p-selectin on their membranes and contains proteins involved in coagulation such as fibrinogen, coagulation factor 5 and von Willebrand factor, as well as protein factor that may be involved in horn healing such as fibronectin, platelet factor 4, PFO, or heparin binding chemokine, platelet derived growth factor, and transforming growth factor beta. Dense or delta granule contain adenosine diphosphate, adenosine triphosphate, ionized calcium, serotonin, and epinephrine. So platelets have two types of granule: alpha granule and delta granule. Alpha granule contains all these protein involved in coagulation like fibrinogen, coagulation factor 5, ponerin factor, and wound healing protein factor like fibrinogen, platelet factor 4, platelet derived growth factor, transforming growth factor beta, etc. And dense granule contains adenosine diphosphate, ADP. D for ADP, D you can see in D is present in dense granule. ATP, ionized calcium, serotonin, and epinephrine. These neurotransmitters are also present in platelet. Epinephrine, serotonin, ionized calcium, ATP, and ADP. After a traumatic vascular injury, platelets can encounter constituent of the subendothelial connective tissue such as bone Willebrand factor and collagen. We have already seen after traumatic vascular injury, platelets encounter constituent of subendothelial connective tissue and such as bone Willebrand factor and collagen. On contact with this protein, platelet undergo a sequence of reactions that culminate in the formation of platelet plug. Platelet adhesion. Platelet adhesion is mediated largely via interaction between platelet surface receptor glycoprotein 1b and bone Willebrand factor in subendothelial matrix. Platelet also adhere to exposed collagen via the platelet collagen receptor GP1A2A, notably genetic deficiencies of BWF, bone Willebrand disease, or glycoprotein 1b, Barnett's cellular syndrome result in bleeding disorders, attesting to the importance of this factor. You can see here platelet addition and aggregation. Bone Willebrand factors function as addition bridge between subendothelial collagen and glycoprotein 1b platelet receptor. Glycoprotein 1b is present in platelet and bone Willebrand factor. This is subendothelial collagen. Bone Willebrand factor substrate bridge between subendothelial factor and glycoprotein 1b present in platelet. Mm, So deficiency of bone Willebrand factor disease result in bone Willebrand disease. Aggregation is this is about addition. Now, what is mean by aggregation is accomplished by fibrinogen binding glycoprotein 2b3a receptor on different platelet. You can see here congenital deficiency and various receptor or bridging molecule lead to disease indicated in color box. ADP. You can see here ADP induced conformational change. This is glycoprotein 2b3a complex It will result in its deficiency will result in a Glanzmann thrombasthenia. This is fibrinogen. Fibrinogen causes bridging of different two different glycoprotein 2b3a, and it causes addition. Glycoprotein 1b deficiency result in Barnett-Salyer syndrome. Remember these three disorder. Barnett-Salyer syndrome is due to deficiency of glycoprotein 1b. Bone Willebrand disease is due to deficiency of bone Willebrand disease, and glycoprotein 2b3 deficiency causes Glanzmann's thrombosthenia. So platelet rapidly change shape. Platelet rapidly changes following addition, being converted from smooth disc to spiky sea urchin, which greatly increase surface area. This change in income accompanied by conformational change in cell surface, glycoprotein 2b3 that increases affinity for fibrinogen, and by translocation of negatively charged phospholipid, particularly phosphatidyl serine to platelet surface. These phospholipids bind calcium and serve as nucleation site for assembly of coagulation factor. So, phosphatidylserine sorbs bind calcium and serve as nucleation site for assembly of coagulation factor complex. Secretion, release reaction of granule content. Secretion, release reaction of granule content occur along with change in shape. These two events are often referred together as platelet activation. Platelet activation is triggered by a number of factors including the coagulation factor thrombin and ADP. Thrombin activates platelets through a special G protein coupled receptor referred to as protease activation receptor 1, which is switched on by a proteolytic cleavage carried out by thrombin. ADP is a component of dense body granules, thus platelet activation and ADP release begets additional round of platelet activation, a phenomenon referred to as recruitment. So, what do you mean by recruitment? See it again. ADP is a component of dense body granules, thus platelet activation and ADP release begets additional round of platelet activation, a phenomenon referred to as recruitment. ADP acts by binding two coupled receptor P2Y1 and P2Y1 to 12. These are given in latest 10th edition. This is not present in the 9th edition. These are latest edition. Activated platelet also reproduce prostaglandin thromboxane A2, a potent inducer of platelet aggregation. Our understanding of biochemical pathway involved in platelet aggregation has led to the development of drug with antiplatelet activities. So, what is significance of studying all these biochemical things to develop drug? Aspirin, the oldest of all, inhibits platelet aggregation and produces a mild bleeding defect by inhibiting cyclooxygenase of platelet enzyme that is required for thromboxane A2 synthesis. This is very, very important in your multiple subject, pharmacology, pathology. More recently, drugs that inhibit platelet function by antagonizing PAR1 or P2Y1 to have been developed. All these antiplatelet drugs are used for treatment of coronary artery disease. Platelet aggregation. Platelet aggregation follows their activation. Their conformational change in glycoprotein 2b3a that occurs with platelet activation allows binding of fibrinogen, a large bivalent plasma polypeptide that forms bridges between adjacent platelet leading to their aggregation. Predictably, inherited deficiency of glycoprotein 2b3 result in a bleeding disorder called Glanzmann thrombasthenia. So we have seen already. Initial wave of aggregation is reversible, but concurrent activation of thrombin stabilizes the platelet plug by causing further platelet activation and aggregation, and by promoting irreversible platelet contraction. Platelet contraction is dependent on cytoskeleton and consolidates the aggregated platelets. In parallel, thrombin also activates fibrinogen into insoluble fibrin, cementing the platelets in place and creating the definitive secondary hemostatic plug. Entrapped red cell and leukocyte are also found in hemostatic plug. in part due to the adherence of leukocyte to P-selectin expressed on activated platelets. So, key concept about platelet addition, activation, and aggregation. Endothelial injury exposes the underlying basement membrane (ECM). Platelet adhere to extracellular matrix primarily through binding of platelet GP1B receptor to bone membrane factor. Addition lead to platelet activation, an event associated with cofactor with secretion of platelet granule contents, including calcium, of cofactor for several coagulation protein and ADP, a mediator of further platelet activation. Dramatic change in shape and membrane composition and activation of GP2B3A receptor. The GP2B3A receptor on activated platelet form bridging cross-link with fibrinogen leading to platelet aggregation. Concomitant activation of thrombin promotes fibrin deposition and cementing the platelet plug in space. Coagulation cascade we will discuss in that video. So this is little bit difficult chapter. The best way to read this is read it again and again. So this is little bit difficult. You may find it a little bit difficult. All these are very high yield topic. So we'll discuss our coagulation cascade in next video. Thank you.